research group that is called the African Computational Genomics, TACG. So today I'll be making this presentation that I've titled Genetic of Cardiovascular and Related Diseases in Africa. If you have any question that time does not allow me to talk about or to comment about, please feel free to send me email. My email is on this slide, or you feel free to just contact me on Twitter. I'm very present on Twitter. So as a way of introduction and background, you may already know that cardiovascular disease, CVD, is a general term that is used to describe the disease of the heart and blood vessel. An example include coronary heart disease, heart attack, and stroke. An estimated 18.6 million people die of CVD every year. This represents 33% of all global deaths, making CVD the world #1 killer disease. So CVD can be caused by a combination of a genetic predisposition and traditional risk factor, such as high blood pressure, high cholesterol, obesity, diabetes, and kidney disease. I think that understanding the risk factor is key to finding some kind of solution to the cure for CVD. Unfortunately, the burden of CVD and its risk factor is increasing in Africa, with expected deaths projected to double in 2030, according to a report by WHO. And given the central importance of Africa to our human origin and disease sustainability, there's a clear scientific and public health need to develop large-scale efforts in examining this disease across diverse populations within Africa. So like you're all aware, genome from African ancestry individual are scarce. Most of the time, when I make these statements, I like to emphasize that human genome data from continental Africa is very, very scarce, particularly when you compare to what we have from the European and other populations. Like you have already heard from some previous presentation, less than 2 to 3 of human genome analyzed so far are being from African ancestry individuals, despite the fact that Africa contains more genetic diversity than any other continent. Again, I always like to emphasize that those 2 to 3% African genome analyzed are mainly genomes of African American individuals. For example, in the past few years, there have been significant advancements in identifying genetic factors that contribute to CVD using an approach called GWAS. And there have been an association of from 100 to 1,000 loci that are known to associate with CVD, but little from Africa. So while we aim to address the genomic imbalance by various efforts in the continent and globally, we as Africans must learn to maximize the use of our rich African data set in the continent. In this presentation, my aim is to demonstrate to you how we the African computational genomes are maximizing the genomic resources in Uganda for novel genetic discovery, polygenic prediction by leveraging strategic regional and global collaboration on genetics of cardiovascular disease, while we also build capacity building and train next generation scientists in Africa. So in order to discover genetic variants that contribute to CVD and its risk factor, my group is using approaches such as GWAS, fine mapping, polygenic risk score, and Mendelian randomization study to infer association, causality, and as well as prediction. So using GWAS, for example, we are interested in identifying what genetic variants are associated with a range of CVD and its risk factor. Once we identify such association, we refine them. We refine those associations as known and null loci. We also use an approach called PRS to estimate individual genetic liability to a disease. A PRS is we calculate the sum of risk allele that an individual has. We weigh them by the effect size as estimates using the GWAS on the phenotype. So ultimately for PRS, we are interested that we may be able to help identify subgroup of individuals in Africa who are at high risk of CVD. So very important approach we also use is called Mendelian randomization approaches. Like you may already be aware, observation studies have shown that there have been an association between CVD and a range of risk factors that I've mentioned, like type 2 diabetes, kidney disease, hypertension, high cholesterol, and the rest of them. So this kind of studies can be limited by failure to adequately control for confounders. So why we use Mendelian randomization study, it will help us to assess the causal relationship between a range of CVD and its risk factors. So first, in Uganda, we set up a genetic epidemiology study using the cohort called General Population Cohort, GPC. GPC is a population-based cohort of about 22,000 people that live in a rural community in Uganda. So this cohort was originally set up in 1989 to examine prevalence of HIV in Uganda population. From around 2010 and 2011, the research activities at CPG was broadened to include epidemiology and genetic epidemiology of non-communicable disease. So every year, our team at the MRC go to the community and recruit people, also measure a range of indices. So we always start by first sensitizing the local community about the study activities. We seek their permission before we go into the community. In the last few years, we have a group that is called a CAB, Community Advisory Board. So this group includes people in the community, very important people in the community, like pastors, like imam, like cultural leader, group leader, as part of our Community Advisory Board, and as well as a professional at the MRC UBRI. So this group, they give us guidance on how to do our research and how to enter into the community. So in this particular study, 5,000 individuals were recruited for genetic epidemiology studies. Another 2,000 individuals were recruited for whole genome sequencing. Yeah, so we went ahead, we collaborated with similar studies in Africa. We collaborated with the ADAM study. ADAM is the African American Diabetic Mellitus. So this cohort is led by Professor Charles Rotimi at the NIH. And this cohort has an individual from Nigeria and Kenya and Ghana. We also incorporated data from the Durban Diabetes Study. That's a cohort from South Africa led by Aisha Motala. And altogether, we have pulled all this data together and have a total of about 14,700 individuals across Africa. So as you can see on this slide, all the study measure a range of CVD risk factors, including HbA1c, which is an indicator for type 2 diabetes. We have a lipid trait, we have blood pressure, we have full blood count, and other traits. So in order to demonstrate the value of African genomes, we published this work in Cell. The project represents one of the largest collection of GWAS and whole genome sequencing in Africa. So as you expect, we found a high level of genetic diversity in the Uganda population than you will see, for example, in the European population. We found specifically 28.7% of SNPs discovered in Uganda whole genome sequencing data have never been found before. This was about 9.5 million novel variants, representing about a quarter of genetic variants identified in Uganda population. So this slide shows the list of novel and distinct genetic associations that were found to be associated with various cardiometabolic traits and disease. So as you can see on the last two columns, this column and this column, with the heading minor allelic frequency for Africa and minor allelic frequency for Europe. You will have noticed that here we have mainly a zero minor allelic frequency, meaning that some of these SNPs are not available in the European population. And this again also emphasizes the need to study African population. And this SNP, which are monomorphic in European population, are found to be associated with a range of cardiometabolic traits, in African population. So this is very, very important. So our study also leveraged on the lower linkage equilibrium in African data set. The fine mapping from our analysis resulted into a narrow credible interval for most of the traits. We were able to map 16 out of 41 distinct loci to a single SNP in the credible interval. We also resolved the previously identified association with HbA1c. So I think fine mapping is an opportunity for Africa. It's an opportunity, particularly when we use the African data set. So the richness of the Uganda genome resource also offers us the opportunity to study the extent to which genetic differences encode differences in traits. And I'll give an example of height, and I'll also give an example of LDL. So we assess the heritability for all the 34 cardiometabolic traits using mixed model approaches that also model environmental correlation. So we found that height, for example, is less genetically determined in Uganda relative to what you will see in European studies. So the narrowest sense heritability for height was 49% in Ugandans, compared to an estimate of about 70 to 80% in the European population. So one reason for this might be environmental factor such as nutrients in Uganda. So in contrast, the heritability estimates of LDL cholesterol were significantly higher in the Uganda population, and that was about 54% compared to 20 to 43% in the European population. So on the whole, our finding highlight the usefulness of examining genetically diverse population within African, and also other populations. So from there, we have now recently extended our collaboration to individual data of African in diaspora. We are collaborating with colleagues on the use of the Millennium Veteran Program, MVP, which also has a range of cardiovascular disease measure. And this cohort has about 57,000 African ancestry individuals. We are also collaborating with data of about 7,000 Black participants in the UK Biobank. So recently, we aggregate all the African ancestry data sets, making a total of about 78,000 individuals of African ancestry. So in this study, we identified more than 400 genetic variants that reach genome-wide significance for diastolic and systolic blood pressure. Of course, most of these genetic associations were driven by the MVP dataset. So Brenda Idosen, who was an MSc bioinformatics student, performed meta-analysis and multi-trait analysis at GWAS to infer a better approach for gene discovery. So in this work from Brenda, so we show that leveraging collaboration between traits like systolic and diastolic blood pressure, it can improve power of genetic association to identify a marker that have not been previously associated. And you can see from this Venn diagram, you see using multi-trait, you have an extra gene discovery that were not found using meta-analysis. So similarly, using the same data set and also using a similar method, but this time around, different traits, lipid traits. So Toure, who is from Mali and also doing MSc bioinformatics, identified 6 novel loci using a univariate method and 10 novel loci using multi-trait method across all the 4 lipid traits. So we leverage the shared information between the traits to perform multi-trait fine mapping using a tool that is currently being developed called FlashFM. So FlashFM is a Bayesian fine mapping method that explores the shared information between those traits, like lipid traits, LDL, HDL, and total cholesterol. So this figure shows comparison of credible set size across loci with multiple association with FlashFM when plotted against a very popular tool called FineMap. So both Brenda and Ture are currently writing up this work for publication. Again, using the same data sets, we assess the performance of lipid trait PRS that were derived from summary statistics from European population and African American, multi-ancestry individual, and as well as a multi-trait approach to understand the transferability of PRS from different populations to African population. So the S-axis, for example, show different ancestry. Like I've mentioned, you see here is blue, here is the African population. You see the MAA here is the multivariate where you jointly assess the different lipid traits. So here we show that African American derived PRS were more predictive compared to multi-ancestry and other PRS. And this is very important because current publication and the correct concept is that using multi-ancestry derived PRS is enough to predict into any population. So notably also from our study, we see that using the multivariate derived African American PRS was even more predictive than univariate approach of multi-ancestry and as well as the European population. Here you see the African population predicting far better. You see the multi-trait predicting much better before the multi-ethnic population, which is currently being said to predict better in all populations. So our result is a total divergence from the PRS plot that you are familiar with. Many people are familiar with this plot from a paper that was published by our friend Alicia Martins, showing a poor predictivity into African population. From our study, you will see that African predicting better because we are using an appropriate data set. So again, our study suggests that improved representation of African individuals will ensure generalization of findings for precision medicine. So in another study from our group, which this work, for example, currently is under review, we show that the African American derived PRS was more predictive of type 2 diabetes compared to multi-ethnic derived score. So this finding, it was from one of the cohorts from the Zulu cohort in South Africa. It was also replicated in the Adam cohort from Nigeria, Ghana, and Kenya. So again, so this seems to be a contradiction to previous study, which suggests that PRS based on data from multi-ancestry population improve prediction for all population. So most important from this study, we found that the participants in the 10th decile of the PRS had more than 3-fold greater risk of developing diabetes, and they were diagnosed 2.6 years earlier compared to those in the first decile. We are now expanding this type 2 diabetes study in the work of Chisom Shuremekun doing a PhD at Makerere University in Uganda. So Chisom is looking to create the African ancestry genomic architecture of type 2 diabetes. So she aims to include the evaluation of transferability of PRS in different regions of Africa. For example, we want to know how to PRS transfer between the West, East, and the Southern Africa. So in a pilot work, she meta-analyzed type 2 diabetes viewers in 3 studies, the Adam cohort, the South African Zulu cohort, and MVP, and found 4 distinct associations. So she's now exploring how best to find map to find map the novel loci and as well as the no loci of type 2 diabetes. So using the result from meta-analysis of lipid traits and type 2 diabetes from both Ture, Chisom, and Brenda, and other people from the group. So recently, we investigated the causal effects of type 2 diabetes liability and lipid traits on the risk of stroke in African ancestry individuals. We compared this to the estimates that have been obtained from the European ancestry individuals. Like you can see on this slide, so we find that higher genetic proxies type 2 diabetes liability and LDL, total cholesterol, and lower genetic prophecy HDL were associated with increased risk of stroke in African ancestry individuals. So we also found that there's no evidence of differences in the estimates. So when performing analysis in the European ancestry and as well as African ancestry individuals. So this work involving many members from our group has now been published in a journal that discusses stroke. So again, our analysis here support a causal effect of type 2 diabetes and lipid traits on stroke in African ancestry individual, which is very similar to what you also see in European ancestry individuals. So considering the high burdens of kidney disease in Africa and Uganda, we're now leveraging the use of a biorepository to measure serum creatinine in Uganda and as well as other kidney biomarker in Uganda. So these resources are very, very important, particularly for sustaining genomic study in underrepresented population like ours. So recently we performed GWAS in 3,000 individuals in Uganda. We identified and validated 2 EGFR loci at HBB and as well as GATM. So as GATM locus, the association signal in Uganda was distinct from previously reported signal and this locus. At the HBB, the association signal has been previously reported, but the lead SNP at the HBB loci accounted for 88% of posterior probability of causality after fine mapping. So this work again was published recently and also again represent the first GWAS of kidney function in Africa. Our findings further highlight the importance of diverse ancestry for uncovering novel association. Even now, so larger continental African meta-analysis are now needed to gain further insight on the genetic architecture of kidney function in African populations. So this is from Richard Mayanga, who is a second-year PhD student at Makerere University in Uganda. Richard is now expanding on our kidney function GWAS work. Richard is focusing mainly on aggregating genomic data set of kidney function from continental Africa. So Richard's work involved expanding the UGR, that's the Ugandan genome resource, from that 3,000 individual that I showed previously, and she's now trying to expand that by meta-analyzing them with other important data sets from Africa, such as the ADAM data set led by Charles Rotimi, the DDS from Aisha Motola, AWGEN data set from Michelle Ramsey, and Meru data set from Mia Cramping. So like you can see here, so the Meru data set is a Brownian quartz for genomic study. So we are currently genotyping up to 8,000 samples on H3 Africa array. So Richard will be using this resource to build capacity in conducting genotype calling, doing quality control, doing genotype imputation, and as well as doing GWAS. So in a pilot study that was done by Richard recently, so Richard meta-analyzed UGR and ADAM cohorts. We found a novel loci at chromosome 1, which was implicated in a gene called PDZK1. So using GTES and other gene expression resources, so we found that PDZK1, a protein coding gene, is highly expressed in kidney and liver. So we think this is a very interesting discovery for kidney function, which we're now presenting here for the first time. So Richard is going to be working more on this to investigate this further. So Richard is aiming to publish this work after aggregating more data set from AWGEN, from DDS, and as well as Meru quartz for his PhD work. So as part of our investigation to understand kidney function in African population, so Miriam, who is a Richard Fellow in our group, is working to determine the frequency of 2 ApoL1 risk allele, G1 and G2 in the Uganda population, and also trying to investigate the association with kidney function, blood pressure, and bilirubin level. So this slide, for example, shows high frequency of 1 or 2 copies of ApoL1 risk allele in West Africa, in Nigeria, Gambia, Sierra Leone, than you will see in the South Africa and West Africa. You will see here the blue color showing no risk allele, and so yellow color showing 1 or 2 copy of the risk allele. And you can see a pattern of high frequency of 1 or 2 copy of risk allele in the West African population, and you will see a lower frequency in the East African population and South African population. So, and again, this is also consistent with prevalence of kidney disease in different regions. So whenever you see high prevalence of kidney in African population, we also see high frequency of 1 or 2 copy of the risk allele. We think this is a very good pattern, and Miriam is looking to write this up after further analysis. So in our recent published work on GWAS of kidney function in the Uganda population, so we reported that multi-ethnic based PRS do not predict well in the Uganda population. So Chris Kintu, another PhD student at Makerere University, is developing a PRS score for kidney function from diverse population, and he's trying to evaluate the transferability of a kidney function score in multiple ethnic groups in Africa. So this genetic score, when developed, can be used as instruments to assess whether cardiometabolic traits and disease are causally related to the risk of chronic kidney disease in Africa. And Okwe, who is a postdoc in our group, is leading a bidirectional two-sample Mendelian randomization study using EGFR as both exposure and outcome to investigate the causal relationship between each cardiometabolic trait and kidney function. So just to summarize everything I've been saying, so most large-scale study of genetic of CVD and its risk factor, they have been dominated by participants of European ancestry. So therefore, African ancestry population currently lag behind. So in our research group at the AfSHG, so while we aim and we are also looking for a way to address the genomic imbalance, so we are using approaches such as GWAS, fine mapping, polygenic risk score, and Mendelian randomization study for gene discovery, for polygenic prediction, and also investigating the causal effects of modifiable risk factor on cardiovascular disease in Africa. So our ultimate aim is that we might be able to find insights that will guide the prevention and treatment of CVD in Africa. To conclude, I would like to thank all members of our group who have done most of the work that I've presented today. We have Okwe, who is a postdoc that is doing very well. We have 4 PhD students, 3 of them are registered at Makerere University. Sarah Silva registered at the London School of Hygiene and Medicine in the UK. We have a PhD student and also a research fellow that have been making a lot of contribution. I also like to acknowledge our friend, our partner, that are also contributing to our study in the group. And most importantly, we have been very, very fortunate to collaborate with a range of leading research groups in Africa and globally. Particularly, I would like to acknowledge Dr. Charles Rotimi's group in NIH, Aisha Matala in South Africa, Michelle Ramsey, Mia Crampin in Malawi, and others. So my group has also received a lot of mentorship from these people, including Moffat Urenda at the MRC, Uvira in Uganda, Andrew Morris in Manchester, Monica Woody and Derek Widman at the University of South Florida, and as well as Stephen Borges at the University of Cambridge. So finally, I would like to acknowledge the contribution of the consortium, the Asia Africa Bioinformatics Network, PTS's team at the Asia Africa, CKD Gen Consortium, and the Cogen Kidney Consortium. So thank you very much, everybody. Just before I go, I would like to say that we are now looking to expand our group and looking for a staff scientist who have experience in doing GWAS, fine mapping, PRS, and Mendelian randomization. We are looking for somebody that can be based in Uganda or Malawi, or somebody that can be based at home in their pajamas and do the work. So if you are interested, look for somebody that can start either in December or January next year. Please feel free to contact me. Thank you very much, everybody. God bless. Bye-bye.